some marine natural product like antibiotics, enzyme,
Excuse
alkaloid.
me, ma'am.
Yes.
Ma'am, sorry to interrupt, but I want to ask, like in report, we need to mention each and every word because everything is new for us to study, whatever we study here.
Yes.
So report is exceeding so many pages as per your syllabus. So can you tell what can we do in that case?
1 second. Okay. Okay. So, what can I say? Actually, I try to cover many topics so that you should get the idea about new things. So, Sharat sir, can you help out?
Yes, ma'am. Report can be exceeded as many pages as they are willing to prepare. It's
Okay.
not like they can just compile it within 50 pages. It
Yes.
could exceed even 500 pages. There is no limit to reports pages. So
Okay.
it's upon the capability of the intern how much pages they are able to prepare. It's
Okay.
that it shouldn't be less than 50 pages. It is that
Okay.
not like it cannot exceed 500 pages even. It could exceed 500 pages even if they are
Okay.
capable of
So that
preparing a
we that would be their PhD thesis then.
500. That is the thing.
Yes. Okay. Your problem solved.
And ma'am,
So,
plus, like definition is going to be similar. Whatever you write, same we will write here. So there is nothing going to be different in our report and your PPT. Like judgment and all such things, how it's going to be performed that way. Diagrams is also going to be same,
not
definitions, procedure. Like,
necessarily. Yeah, you can say that. But don't, you can paraphrase or rephrase that also. The topic, you can paraphrase that.
okay, ma'am.
Okay. So don't worry. There are some terms that that for sure will be same only. So you can paraphrase or rephrase that. Yeah. Okay, there is one announcement. I'll share that message to sir as well. So we are planning like today is our 7th day. Okay. So 1 second. Huh. So on Saturday, if we think that Saturday, Sunday, I will take you a lecture, and for 2 days there will be your assignment and report submission. So that will be a continuation thing. We will complete our 10 days syllabus as well, and you will get chance to cover everything in your report. At the same time, assignment parameters will be very easy to judge for me. So on Saturday, Sunday, I'll conduct the lectures. So timing, you say if you are comfortable Saturday, Sunday, you just suggest the timing, sir.
Ma'am, on Sunday, there is already a program lined up that is
Okay.
at 6 p.m. So, if it is feasible to conduct during morning hours like 11 a.m. or
On
12
Sunday,
p.m. on Sunday,
yes, yes, no issues.
like at 11 a.m. or 12 noon, we
Yes.
can conduct.
So we can do one thing. We will conduct our lecture on Saturday at the same timing, 6 to 7, and on Sunday that will be 11 to 12.
Yes, yes, yes. 11
Okay.
to 12 or 12 to 1, whatever it is
Yes,
feasible.
11 to 12 will be okay.
11 to 12 will be okay.
Yes. So I'll mention the timing as well. So it will be a continuation for us as well. At the same time, you can compile your report, you can prepare your report covering all the aspects. So there will be no off for Saturday, Sunday. Your preparatory leaves or assignment leaves will be on Monday and Tuesday. Yes. Okay. Then we had started dealing with the bioactive compound from marine organisms mechanism, after that sources. Then we took the example of some marine bioactive compounds, then their application in various fields, then marine derived biomaterials, what biomaterials are, then what are the properties, which property ideal biomaterial should carry, then their application. After that, we discuss certain important marine biomaterials that are usually used in different aspects, then summary chart. After that, we had started with introductory part of our different marine bioactive compound, like antibiotic, enzyme, alkaloids, biomineral, biocomposite, and biopolymer. From today, we are going to deal with each and every biomaterial or bioactive compound in detail. First is antibiotics. You all know that. Okay, is there anyone who can tell me the discovery of antibiotic?
The penicillin.
Yes, first antibiotic was penicillin only, Penicillium notatum. Yes, Jeet.
The first antibiotic that was discovered, the first naturally discovered antibiotic, it was Penicillium notatum. But before that, also there was a compound that is salvarsan that was discovered during the time of the World War, and this was utilization of arsenate because in those days people were not much aware of its toxic effect. So that was a synthetic antibiotic that was first discovered.
And what about natural antibiotic discovery?
Penicillin from Penicillium notatum. It
Do
was
you
a
know
fungus
any
which was,
historical? Yes, it is a fungus. And how it got discovered? Do you have
and
any
also
idea?
it was an accidental discovery because
Yes,
Alexander Fleming was working with Streptococcus species, and then he left that petri plate open and went out for some holiday or like that, and after that he found a mold that was growing on the plate, and it inhibited the growth of the Streptococcus. But although he could not coin the term antibiotic or say that this is exactly antibiotic, he said that there must have been some compound that this organism is releasing.
yes,
Later on, it was worked on and it was discovered as an antibiotic.
yes. So, what are antibiotics?
Antibiotics, as the name suggests, it means against life. So it is any compound naturally derived or synthetic or also semi-synthetic that is naturally derived and later on chemically modified. And
Yes,
it is effective against microbial organisms, against
yes,
bacteria, virus, protozoa, this type.
yes.
And they are named accordingly, like antiviral, antiprotozoal, like that.
Exactly. Very good. So we will start with our topic antibiotics. There are certain marine organisms that used to synthesize or produce certain antibiotic. Antibiotics are nothing but these are the bioactive compound that inhibit the growth of other bacteria. Either they will inhibit the growth of bacteria or they will kill them. By two ways they used to work. So antibiotics, these are nothing but the bioactive compound that inhibit or kill the bacteria, inhibit the growth of bacteria or they kill them. At the same time, antibiotics are playing an essential role in the medicinal field to treat certain bacterial infection. When we talk about biomaterial, antibiotics are often incorporated into materials to prevent the bacterial colonization and infection. That means bacteria, their growth will be inhibited.
वहाँ नहीं जाएगी क्या?
What happened? Whose mic is on?
बस आगे भी.
Okay. So, if we deal with the antibiotics, they are very important for medical devices. They are very important in implants, then wound dressing, where the risk of infection is high. Okay. So there is one interesting field, Sanskriti. Okay. So in wound healing, like you all know the dressings. Nowadays, Sanskriti, your mic is on better. Okay. So when we talk about wound healing, there are certain wound dressing material that used to. I think 1 second. Okay. So there are certain, we can say, wound dressing material where we can incorporate this biomaterial, and in the wound dressing itself, they will play a antimicrobial, antibacterial role. So this is also an emerging technique nowadays in tissue engineering, where we are also working in our labs. So bioactive compounds are naturally occurring molecule that exert a biological effect on other organisms. So antibiotic is nothing but a naturally occurring bioactive compound, and that is responsible to exert or to implant certain biological effect on the bacteria. Marine organism, as we all know, they live in extreme condition. They live in competitive environment as well, where pressure will be high, salinity point of view, there are certain organisms that live in extreme saline condition, then extreme temperature they can tolerate. So they might be I'm using the term because there are certain bacteria antibiotics that yet to discovered. So they are very useful, or they might be useful for the production of bioactive metabolite, including antibiotics. So marine biotechnology, they uses these resources, and these marine bioactive compounds will be useful in the drug discovery, will be useful for the biomaterial development, and in health point of view. Now, there are certain marine sources of antibiotics. Marine microorganisms, they are highly having capability to synthesize the antibiotic that are novel in their own. And why they are novel? Because their metabolic pathways are very different. These are not synthesized by a terrestrial organism. Major marine antibiotic found is marine actinomycetes, that is synthesized by marine organism, that is Salinispora. Then there are certain marine bacillus species. Then marine fungi there, Penicillium aspergillus. Cyanobacteria are there. They are also having good capability to produce antibiotic. Marine sponges, marine macro and microalgae, then marine tunicate. These are all the marine organism. These all are having an property to synthesize a novel antibiotic. So these are the example of marine antibiotics and their sources. After that, I have also mentioned some example of marine derived antibiotics that are widely used. Mentioned the compound, after that source organism and activity. The first source organism is Salinispora tropica, and this Salinispora tropica used to synthesize a compound. Name itself is on the basis of genus, that is Salinosporamide A, and it is having good activity against certain bacteria, and it is also anti-cancerous in nature. After that, Streptomyces species is there. Marinopyrrole A antibiotic or compound is synthesized by this, and it is active against MRSA. This is the methylene resistant Staphylococcus aureus. Again, bacterial infection, for the bacterial infection caused by this organism, this compound marinopyrrole A is used. After that, there is bromophycolide A. This antibiotic compound is synthesized by algae, that is Caloficus serratus, and it is also having good antimicrobial and antifungal property. After that, there is species Pseudo Alteromonas, and it is responsible to produce thiomarinol, and it is a broad spectrum antibiotic. So broad spectrum antibiotic, when we talk about this antibiotic is not specific to one organism. It can be applied for several antibacterial aspects. Then marine sponge is there, that is Dragmacidon, and it is used to synthesize Dragmacidin D, and it is having property that is antimicrobial, antiviral in nature. So again, I'm repeating. We have discussed about marine derived antibiotics. Antibiotics are nothing but the bioactive compound that will ultimately inhibit the growth of bacteria or kill them. There are certain bacteria like Salinispora tropica, Streptomyces species, Caloficus serratus, Pseudoalteromonas, marine sponge that is Dagmacidon. They will synthesize certain antibiotic compound like Salinisporamide A, marinopyrrole A, then bromophycolide A, thiomarinol, then dragmacidin D. These are all certain antibiotic compounds, and they will show certain activity against bacteria. After that, this marinopyrrole A is used to treat, or is used to uh, in the drug or in the medicine that is caused by the Staphylococcus aureus. After that, bromophycoloid A shows antibacterial, antifungal property. Then Pseudoalteromonas species used to synthesize thiomarinol. This is the antibiotic compound, and it is having a broad spectrum antibiotic. And last one is marine sponge. These are some example I have mentioned. That is Dragmacidin D compound, and it is also having good antibacterial and antiviral property. After that, next is mechanism of action of marine antibiotics. Now, when we talk about bacteria, their outermost wall is nothing but cell wall. So there are certain parameters how they will act on the bacteria and how they will kill or inhibit the growth of others. So just go through the things, and if you have any doubt, you can ask.
Hello, ma'am.
Yes.
Ma'am, if we are using antibiotics, if the antibiotics work is done by the certain that product,
What?
then afterward, what happened, ma'am? Means the remaining effect of the antibiotics.
See, this is a general question. It is not marine biology specific question, but I would like to give answer. When we take excessive medicines, ultimately you will get used to that. Just take the example, like if you anyone is taking sleeping pills. So what happens? Uh, Initially, that 20 mg will affect in a good way. Okay, but after some day, after 1 month, this 20 mg will not affect in our body. We have to raise the bar. 20 to 50, 50 to 100, 100 to 150. This is the case with antibiotic. And uh, there are certain doses on the basis of your infection. Beyond that, there is no need to take antibiotics till the infection get inhibited or infection get eliminated. Till that only you have to take that medicine. Excessive dose ultimately cause infection or ultimately damage your natural metabolic pathways. That's why doctor used to suggest this is for 3 days only, this is for 5 days only. So this is the parameter. Okay. Okay. Then Marine biomaterial for antibiotic, mechanism of action of marine antibiotic. When we take antibiotic, how it will kill the bacteria or how it will inhibit the growth of bacteria. First, they will inhibit cell wall synthesis. When bacteria used to replicate or multiply, the basic component is their cell wall. When we inhibit that cell wall formation, there will be no further proliferation. Second is you can damage the cell membrane of that bacteria. So they will get inhibited or killed. In every cell, there is essentiality of protein synthesis. So we can inhibit that protein synthesis. We can inhibit the DNA or RNA, that is genetic material, and their replication. At the same time, if we talk about viruses and fungi, antiviral antifungal effects in some compounds, these antibiotics can also work for this. So these antibiotics, they often show novel chemical structures, and that are again, I'm just mentioning the importance of marine organism. Terrestrial organism cannot synthesize these novel antibiotics, and that's the reason where we have to do deep research related to marine antibiotics. Then marine biomaterials for antibiotic delivery. Marine derived polymers or antibiotics, they are used as a carrier for antibiotic delivery. So antibiotic, there are certain polymers, when monomer combine, they will form a polymer like carbohydrate, protein, fats. These are all polymers. These polymers are used as a carrier because these polymers, they show a good biodegradability. They are very compatible with our cell. They will not damage our cell. They show good antimicrobial properties. Now, my first question is, when we take antibiotic, why these antibiotics will not cause harm to our cell? When they are killing bacterial cell, why they are not killing our cell? Just raise your hand. Don't answer openly. Any other answer? Jeet?
The reason for which the antibiotics were in the previous times also until many date, it is also known as the magic bullet. It is basically targeting those parts of the cell which are specific to the bacteria, like the bacterial cell wall. In our case, we have no cell wall, and in the bacterial cell, they have that peptidoglycan layer, which it basically targets, and the peptidoglycan binding protein to which it will bind, as the result of which the interlinkage between the peptidoglycan layer, that is the pentaglycine interlink, that will be inhibited. As a result, the bacteria will get killed. But in human cells, we do not have that peptidoglycan layer, so there's no way of affecting the human cell.
Yes,
And as a
exactly.
result of it, yes,
Yes.
the cell wall affecting antibiotics, they have higher efficacy than those DNA binding antibiotics, as the DNA component is almost same in case of
Yes.
the prokaryotes and eukaryotes.
Yes, exactly. So they affect the cell wall, they affect the bacterial cell membrane, and we our cell do not carry any cell wall. We are all eukaryotic cell, and where cell wall is absent. Okay. Now, some common biomaterial. Just read first line. There are certain biomaterial that are very essential to carry this antibiotic delivery. And common biomaterial are chitosan, alginate, carrageenan, marine collagen. Chitosan, this is also used in tissue engineering as a base substance. So, one second. Chitosan is produced or synthesized or formed from crab shell, shrimp. It shows antimicrobial activity. At the same time, it is also useful in the wound healing creams or bandages. Alginate, that is also obtained from the brown seaweed. Weeds are unwanted plant and they form hydrogel. They are also used in wound dressings. They will absorb all the moisture. Then carrageenan, it is synthesized by red algae and it is used for the drug encapsulation, that drug coating. In drug coating also you can use this carrageenan. Then there is marine collagen that is formed or produced or extracted from the fishes, and these are used in the tissue scaffold. After that, there are some ways antibiotics are used in the biomaterial, like in the coatings. Antibiotics can be incorporated into coatings applied to the surface of biomaterial. So whichever biomaterial you are going to implant, like just take an example of rod. If I'm implanting that rod in my body, that rod should be coated with the antibiotics of this marine organism, and this coating will release antibiotic slowly over time, and it will give the durability of that rod, and it will show their long lasting protection against certain very microbial infections. Then second is scaffold. Yesterday I have discussed or mentioned about the scaffold. It is the framework. So that scaffold where cell will take the guidance how to proliferate, how to grow, that scaffold can be incorporated with this antibiotic. Again, it will just prevent the infection during the proliferation or regeneration of new cell or tissue. Next is implant. Like there are certain implantation. So in the implantation, these implants or whatever you are going to insert in your body, you can insert the material or equipment that is getting inserted, that should be coated with the help of this antibiotic synthesized by marine organism. And next is wound dressing. Wound dressing can be coated or can be dipped in some antibiotic, and after that in a uncontaminated region or without any contamination you can wrap it up, so it will also prevent or treat the infection. But there are certain current challenges also. Many marine organisms that are showing this or that are producing or showing their capability in the antibiotic field, they are unculturable with current techniques. So it is the current challenge where we have to grow or we have to provide the favorable condition in the lab. At the same time, okay, what are metabolites? You can give answer. What are metabolites? Okay. Metabolites, yes.
Metabolites are basically small molecules that are, we can say, product of metabolism, the like various chemical processes which are occurring in living organisms.
Yes. And types of metabolites?
Ma'am, it can be of various ways, like primary metabolites, secondary, which contains vitamin, amino acid, like this.
And what is the basic difference between these metabolites?
Ma'am, it can be like complex on the basis of complexity, we can say, and the function they perform, like signaling, interaction, and environmental purposes related to that.
Okay, not actually. Okay, I'll
Yes.
explain that. So metabolites are the compounds that are released by certain organism. This is the basic definition. Now, metabolites are differentiated into two parts: primary metabolite and secondary metabolite. Primary metabolite are those metabolites that are essential for the growth and development of that particular organism. But secondary metabolites are not essential for the growth and development of organism, and there we are interested in. Secondary metabolite, these are released by this organism and they are not contributing in the growth and development, so we can extract that secondary metabolite for our use. So these are the metabolites. Then next is, one second. These metabolite or antibiotics are nothing but metabolite. They show very less yield. So to enhance the yield of antibiotic, it is a big challenge. After that, resistance development due to overuse or misuse. Like I explained you before 10 minutes, I think, that when you take some medicines, initially that 10 mg also works. But if you're taking that medicine twice a day, every day, it will be not that much efficient, and one day will come that 10 mg will not affect at all. So that resistance development is a big challenge. After that, sustainable harvesting to avoid damaging marine ecosystem. Our marine ecosystem is very unique, so we have to extract all this product without harming the ecosystem, so that our future generation should also get the benefit of this marine ecosystem. So this is also a big challenge. So it is all about our bioactive compound and biomaterial, that is antibiotic. We discuss about what antibiotics are, where they are useful, how they are useful. After that, bioactive compounds. These antibiotics are naturally occurring molecule. Marine organism that show their unique property. They are having their ability to produce unique bioactive metabolites, including antibiotic. And these are very helpful in the drug discovery, biomaterial development, and health application. After that, we had discussed about the marine sources of antibiotics, which are the sources. So, actinomycetes is there, bacillus is there, fungi is there, cyanobacteria, sponges, algae, tunicates. After that, we have discussed about the example of marine derived antibiotic, some compound source and activity we had studied. Then how this antibiotic will inhibit or kill the bacteria. Either they will inhibit the growth of cell wall synthesis, or they will disrupt the bacterial cell membrane and it will ultimately kill the cell. They will inhibit their ribosomal action, that means protein synthesis. They might inhibit the genetic material replication, DNA or RNA. Then they also show antiviral, antifungal effects. After that, marine biomaterials are there that are used to deliver these antibiotics. And some common marine biomaterials we had discussed, like chitosan, then carrageenan, marine collagen, and alginate. After that, where antibiotics are used in biomaterial, so for the coating purpose. After that, in the scaffold, during implantation, wound dressing, we can use this antibiotic. And we have discussed about the current challenges. Which are the challenges? This is all about antibiotic. And our second bioactive compound or biomaterial is the enzyme. Now, what are enzymes? So enzymes are biological catalysts that can enhance the rate of chemical reaction. That means accelerate the chemical reaction. If we are talking about biomaterial, enzymes are used as they are highly specific and they are highly efficient. And when we use enzyme in any process like fermentation process, we can speed up the rate of reaction. That means if my fermentation requires 6 months, with the involvement of enzyme, I can reduce this 6 months to 3 months. That means my production cycle just get double. I will get double benefit. So therefore, enzymes are widely used. Now, enzymes are very important as they are highly specific. At the same time, they are very helpful in designing and functionality of biomaterial. Whichever biomaterial you are going to use, that enzyme will ultimately enhance the property of biomaterial, and therefore it is applicable in medicinal field, biotechnological field, and material science. Now, these enzymes, we're talking about enzymes related to marine biotechnology or marine microbiology. So, I have mentioned the definition. Enzymes, these are derived from the marine organism, and they are having unique property, like they will show salt tolerance to wide range, they will be stable in different temperature, and they are also showing good barophilicity. Now, what is barophilicity? It is the ability of organism to tolerate high pressure as well. So usually this pressure is found in the deep part, deep sea. At the same time, they are very efficient in the lowest temperature as well. That means cold activity, they show a good cold activity. So again, I'm repeating this part. What are enzymes? These are the biological catalyst. What they will do? They will accelerate the chemical reaction. When we talk about biomaterial and enzyme, these enzymes are used because they are highly specific and highly efficient. Enzymes, they are very important in the biomaterial as well, as they are applicable in medicine, biotechnology, and material science. Enzymes derived from the marine organism, and they show unique properties, like they are highly thermostable, they are showing barophilicity, cold activity, and they show a good salt tolerance level. Now, there are certain sources of marine enzyme, mainly marine bacteria like Bacillus, Pseudoalteromonas, Vibrio, marine actinomycetes. These are all the organisms, and they show a good enzymatic production. There are certain fungi like marine fungi, marine algae, marine invertebrate, deep sea extremophil. Extremophils are those that survive in extreme condition. What are halophils? Okay.
Ones
What
that
else?
thrive in the salt concentration, more salt
Yes.
concentration.
And what about sacrophils? These are organisms that can tolerate or that can survive in extreme low temperature, cold temperature. That means you can say minus 15. So in that condition also there are certain bacteria, and they will release some enzyme. And here we are going to use them. After that, I have mentioned about types of marine enzyme and sources. Enzyme protease, function is protein hydrolysis. Then source, from where this enzyme we can take fish viscera and bacillus. Then for the source, deep sea fungi, fish gut, they will release lipase enzyme. Name itself suggests that for the lipid breakdown, after that amylase enzyme is there that will break the starch, and source is marine bacteria. After that, chitinase is there that will be synthesized by actinomycetes, and that will degrade chitin. So enzyme, basically enzyme name itself suggests their function. Cellulase, there will be degradation of cellulose, and source will be seaweed associated bacteria. Then lacase, and this lacase is synthesized by marine fungi, and it is helpful in the oxidation of phenolic compounds. So these are some enzymes, their function and their source. After that, which are the unique characteristics of enzyme? So first is they are active in extreme condition. Like yesterday, someone discussed with me that enzymes are highly temperature specific, and I told you that that's the reason we are going to marine organisms enzyme, because they are active in extreme condition. Second, they are also helpful as they are efficient in their biocatalysis. So they will be efficient biocatalysts, they will enhance the rate of reaction. At the same time, last line, often more stable than the terrestrial enzyme. That's the reason we are going to choose the marine enzymes. After that, where these enzymes are useful. So in wide range like industry, waste management, pharmaceutical, bioenergy, environment, like yesterday, I think Jitu asked about the bioremediation process. So there are enzymes. These enzymes are useful to bioremediate the complex material into simple one. So in detergent industry, food industry, textile industry, this industrial biotechnological perspective uses this marine enzyme. After that, for the waste management, marine chitin, chitosan, these are the product. And for the chitin, chitosan processing, we will use enzyme. After that, pharmaceutical, for the drug manufacturing, for the enzyme based therapies, we can use this enzyme. Then there are certain marine organisms enzyme, they will synthesize fuel, biofuel production. And for oil spill bioremediation, there is involvement of enzyme. Now, how enzymes are used in the biomaterial? First is biocatalysis. Now, enzymes are used in biomaterial as they show a biocatalysis process where they speed up or accelerate or facilitate the biochemical reaction. Like there is one example I have written, enzyme lipase protease. They can modify the surface property of biomaterial or they can degrade the unwanted component. So these enzymes we will add to our product or biomaterial, and this biomaterial will more efficiently work on the degradation process. Second is degradable material. Now there are certain enzymes that can be used to degrade the biomaterial, and like yesterday I told you, we will insert the scaffold which is biodegradable. So nowadays in scaffold we will use enzyme. After giving framework to cell, they will proliferate, and now there is no need of scaffold in our body. So at that time, this enzyme will biodegrade the scaffold in our body, and via excretion, all the material of scaffold just get excrete out. So this is also another aspect. Then bioconjugations. There are certain enzyme bioconjugate. They will attach biomolecule to biomaterial. So enzyme is responsible to attach biomolecule to biomaterial. So that bioconjugation, to conjugate, to attach something to someone, mediator is enzyme. And this can be used to immobilize protein or other molecule onto biomaterial. So protein binding, for protein binding, we will use these enzyme. Enzyme is responsible for binding also. After that, diagnostic devices, there are certain biosensors, and they will detect specific molecule with the help of enzyme. So biosensor will be having enzyme, and this enzyme will detect that particular molecule. After that, wound healing, we can use this enzyme like collagenase is there. It is very essential for healing perspective, and it will just break down excess of collagen in the scar tissue, and this collagen will again be responsible for the wound healing or wound closure. So enzyme collagenase will break down the collagen, and this collagen after breakdown, it will promote the remodeling, that means reformation of the tissue and wound closure. Now, what are the challenges and future prospects? It is very important challenge. There are many organisms that are non-culturable, unculturable in lab. So, cultivation of these microbes is still limited. After that, yield is the major issue when you deal with the marine organism and their byproduct. Then metagenomic study, protein engineering, synthetic biology, their advances is a big challenge. How they will be useful in the enzyme discovery, how you can apply your enzyme in various applications. So that is also a big challenge, because it is considered that there are many organisms enzyme that yet to discover. Study is the another aspect. So this was the second bioactive compound. Now third bioactive compound and biomaterial is alkaloid. Now what are alkaloid? Alkaloids are naturally occurring nitrogen containing compound. Now, alkaloids are very essential or very useful in the pharmacological activities. How? I will explain later. Alkaloids are mainly produced by some marine plant, fungi, marine organism, and they are basic in nature, alkaline in nature, name itself suggests, bitter in taste, and as they are having nitrogen, that means amino acid metabolism is must involved. So what are alkaloid? These are the naturally occurring nitrogen containing compound. It is used in pharmacological activities. Alkaloids are produced by variety of organism, and they are basic in nature, bitter in taste, and they are derived from amino acid metabolism. When the organism will metabolize amino acid, it will release these alkaloids. Now major sources are sponges, tunicate, that is ascadian, soft corals, marine fungi, marine bacteria, marine mollusk, and at the same time there are certain symbiotic microorganisms also, and they also released these alkaloid when they are present in the symbiosis. After that, example of marine alkaloid, there is alkaloid that is Phascaplycine. Major spores is Phascaplycinopsis sponge, and it is having antibacterial, anti-cancerous activity. After that, Haliclona sponge is there. It will synthesize manzamine A alkaloid, and it is malarial, anti-cancerous, and anti-HIV. So in malarial treatment or in that drug, we will use this manzamine A. After that, ascidian, that is eudistoma. Eudistomine C will be synthesized, and it is having antibacterial property. After that, dragmacidone sponge is there. It will release alkaloid, that is dragmacidine, and it is having antiviral, antimicrobial activity. And last example I have shown you, that is from the Aplycina sponge. It will be responsible for the production of alkaloid, that is bromotyrosine derivative, and it is also having antibacterial and antifungal property. So alkaloid, what alkaloids are? These are the natural occurring nitrogen containing compound. Various marine organisms, they release this basic alkaline product, which is bitter in taste, and it is formed or derived from the amino acid metabolism. There are certain marine organisms, they will provide us this novel alkaloid like sponges, tunicate, coral, marine fungi, bacteria, mollusk, and some symbiotic organisms are there. Then I have listed some example like Phascaplycine, manzamine, eudistomine C, dragmacidine, bromotyrosine derivatives, and their sources and their activity we had discussed. What about their biological activity? So, these alkaloids are mainly, you can say, essential as they are having wide range with respect to bacterial and fungal activities. So, if you talk about MDR strains, that is multi-drug resistance aspect, these alkaloids are useful. After that, they also show some antiviral properties, and that is against HIV, herpes simplex virus. Some alkaloids are having anti-cancerous property. They might show cytotoxic effect. They often work as or perform apoptosis or DNA interaction. Okay, is there anyone who know apoptosis?
Yes, ma'am.
Yes, ma'am.
Programmed cell death.
Yes, it is the cell death. So there will be series of reaction, and with this reaction, ultimately a bacterial cell will die, anti or any cell will die. Anti-inflammatory activities, anti-malarial potential is also shown by this alkaloid. Now, alkaloids are having diverse biological activity, and they are very useful in the biomaterial. First is biomedical application. There are some alkaloid that show very important pharmacological effect like analgesic, anti-inflammatory, anti-cancer, antimicrobial. You must be knowing about anti-inflammatory, anti-cancer, but what is analgesic? So analgesic is a pain reliever. So you can say that painkiller, in the painkiller you can use this alkaloid. After that, for anti-inflammatory, anti-cancerous drugs, in antimicrobial drugs, or for treatment of certain bacteria, you can use these alkaloid. And because of these properties, in the drug delivery, in the tissue engineering, these alkaloids are very useful. Second is biomineralization. Now, what is biomineralization? The process by which living organism will produce mineral. So alkaloid play a significant role in the production of this mineral by the living organism. And some alkaloid, they influence the formation and structure of biomineral that are very useful in biomaterial sciences, and these mineral properties are will be useful in the drug or pharmaceutical industry. After that, bioaddition. Alkaloid that are synthesized from marine organism, they have found to have strong adhesive property. So in the wound healing, for the tissue repair, you can use this bioadhesive property of alkaloid. Then bioavailability enhancement. Alkaloid can also be useful to enhance the bioavailability of drug. So you can enhance the efficiency of drug, and this drug will be easily accessible, easily available to your individual cell, and they will improve the therapeutic efficacy. Next is last one, biocompatibility. When we talk about biocompatibility, these alkaloids, they are very specific compound, but they are compatible in our cell. There are some alkaloid that have found to be cytotoxic. There are certain alkaloid or examples of alkaloid that are relatively non-toxic, and they are biocompatible as well. So it depends on your aim, which alkaloid you have to take for that purpose. And if they are biocompatible, that means they will be safe to use in the biomaterial, or they will be safe to use to humans. Now, marine alkaloids, they have been incorporated or added in the biomaterial. So in the scaffold, for the wound healing, for the drug delivery system, in the anti-cancer alkaloids will be released. Then in the tissue engineering, for the coating, you can use this alkaloid. Then some examples I have mentioned. Chitosan alginate sponges loaded with alkaloid. So for skin infection treatment, you can use this alkaloid. So major basic component was chitosan and alginate, but alkaloid will enhance the efficiency, availability of this chitosan. Then marine collagen hydrogels delivering anti-cancer alkaloid locally to tumor site. That means when you take medicine anti-cancerous agent. Now, cancer is nothing but massive growth, rapid growth of cell. So, how this medicine will go to that particular region where cancerous cells are present? So, that specificity is given by this alkaloid. Then, in the nanoparticle, this is also very new concept. For the encapsulation purpose, for the targeted therapy, you can use this alkaloid. Now, what is the role of this alkaloid in the marine ecosystem? Sorry. So, alkaloid, they show a good defense mechanism for the sessile organism. Now, what is sessile? Meaning of sessile? Sessile means that are fixed to one place. And this we had discussed in our previous lesson also. Sponges, tunicates are there. Their barnacles are there. They are sessile in nature. After that, marine they help marine organism to detect or to deal with the predator microbial infection. After that, there are some alkaloid. They often produced by microbial symbionts. Some symbiotic association also produce alkaloid, and that also helpful in the marine ecosystem maintenance. After that, in biotechnology also these alkaloids are very important. So, in metagenomics, in genome mining, biotechnological this method discover biosynthetic gene cluster for alkaloid production. So we can use specific gene and this gene will produce alkaloid in labs. After that, marine microbes culturing is a very challenging aspect. But when you get exact fermentation condition like pH, temperature, agitation, aeration, you can produce this alkaloid in lab or in fermenter as well. After that, synthetic biology, with the help of engineering, microbes will be engineered and this microbe will produce marine alkaloid at large scale. After that, AI-based compound screening. That means whichever alkaloid you are synthesizing, their bioactivity, their binding target can be detected with the help of AI. After that, there are certain challenges like low natural abundance. It is very difficult to extract large quantity of alkaloid, and their chemical synthesis is a big challenge. Sustainability concern is very important. Whatever you are going to use or extract, you have to take care of your ecosystem, marine ecosystem. Future directions, when you talk about future prospects, development of alkaloid is a big challenge, and if you get success in this, it will be a boom. It will be very helpful in the infectious diseases and cancer. Use of marine derived alkaloid biomaterial in regenerative medicine. So in the regenerative medicine where alkaloids are getting used, it is a very good future prospect. Marine alkaloid database, their maintenance, their genetic content databases, biosensor platform is also a big challenge. At the same time, it is a good scope and their analysis is very important. So this is all about today's lecture. We had started with antibiotic, second was enzyme, and third was alkaloid. So is there any doubt? Is there any doubt?
Mom,
Yes.
ma'am, are those marine alkaloids are having any wide range of usage in these days?
Everywhere. In everywhere we are using these products better. And I'm means giving you everything that is based on the recent advances only.
Okay, ma'am.
Yes.
like in the healthcare industry as well, no?
Healthcare, cosmetic, you can say pharmaceutical aspect, tissue engineering, everywhere these marine products are being used.
Okay, ma'am.
Yes. Is there any doubt?
Excuse me, ma'am.
Yes.
Oh, ma'am, I want to know generally, like we discussed about antibiotics.
Mhm.
So, apart from antibiotic, if we are having medicine for any particular disease related to any hormonal disbalance or lowering or excess production of that particular hormone, so does medicine really help to overcome and able to cure it fully?
See, what is important is that the completion of that dose. What normally we do, if I am having fever, I will take that medicine till my fever get down. After that, like doctor suggested or give medicine for 7 days. In 3 days, I'll stop medication. That is the major issue, where my fever get down because I have suppressed the activity of that bacteria that is responsible to cause fever. But after some day it will just get inactivated in my body, but after getting favorable condition, because I have stopped taking medicine. So after getting favorable condition or after not getting those antibiotics, this cell will proliferate again and frequently I will get that you can say fever. So this is the basic reason where we should complete our dose.
Hormone is something which we can't say ki, like all these matlab, hormone needs very particular things to secrete. If
Of
we
course.
talk insulin, people taking insulin for sugar purpose, and the thyroid problem is also increasing day by day. So this is related to
I am not
hormone
taking any
basically.
antibiotic for that.
मैम हॉर्मोन्स के लिए वी आर टेकिंग एंटीबायोटिक और hormonal medicines are there ना, about that I am asking to you कि क्या सच में मतलब जो medicines हैं हार्मोन के लिए बनाई हुई वो प्रोडक्शन को हेल्प करती हैं हार्मोन की कि इंक्रीज करेगा डिक्रीज करेगा, कैन इट एबल टू बैलेंस इट?
Yes, yes, yes. That's why you have seen in your home or in your relative, if you gave the example of thyroid, that hormone will specifically work on the thyroid gland, and with the thyroid gland functioning, that will be enhanced with the help of these medicines.
सो मैम इट्स मतलब साइड इफेक्ट्स वगैरह क्या रहते होंगे हार्मोन के केस में?
साइड इफेक्ट्स बहुत है. ओबेसिटी इज द मेजर साइड इफेक्ट. After that, lethargicity. After that, you can say efficiency of the body cell. So these are all the side effects. When you take excessive medicine, side effects will be for sure there.
Ma'am, have you heard about like neurological disorders related to like Parkinson disease? Have you studied about that? So
Partial,
particularly in that case,
actually, but I know the basic principle, and you also know that there are no medicine for this. We can postpone the effect of the things.
so if we are taking any hormonal medicine for that, as we know there are only one or two particular medicine, and still the study is ongoing related to this one. So as we can treat or can able to cure and try it for having healthy lifestyle basically. So
See, in this disease, there is the best way, your lifestyle, your diet. That is the important thing. But if you talk about side effect, again I will say there is no medicine for this disease. You can postpone or delay the things. So whatever effect I will get in 1 month, it will be delayed for 3 months. That's what we can do for some diseases.
okay, ma'am, thank you.
Okay. Okay, then see you tomorrow. I'll just send message to sir regarding our Saturday and Sunday's lecture. Okay.
Thank you, ma'am.
Okay, thank you.
Thank you,
Bye.
ma'am.
Yes, see you tomorrow.
বুঝছো ক্লাস নাই তো হ্যাঁ হ্যাঁ ওই এই দিদা ଟିକେ ଦେଖେ ওই ଟିକେ ଦେଖ ଏ ଯୋଉ କଲ୍ କଲେ କାକୁ এটা তো আসছে দুইটা সিମ୍ ଅଛି ଗୋଟେ ଏୟାରଟେଲ୍ ମୋର ଅଛି ଏୟାରଟେଲ୍ କରିବି ହଁ